my pleasure to introduce Scott Linderman today. Scott does really interesting work at the intersection of machine learning and computational neuroscience. Scott's currently a postdoc at Columbia, working jointly with Liam Paninsky and Dave Bly. And before that, he got his PhD from Harvard in computer science, working with Ryan Adams and Leslie Valiant. Before that, he was at Cornell in electrical and computer engineering. But in between undergrad and grad, he took some time here in Seattle working at Microsoft, so he's familiar with a lot of our haunts around here. And Scott's been awarded lots of different fellowships and awards, including the prestigious Leonard J. Savage Thesis Award and an AI Stats Best Paper Award. And today, Scott's going to be talking on machine learning models and algorithms for neuroscience.
Wow. All right. Thank you so much. It's really great to be here. As Emily said, I actually lived just a mile and a half down the Burke-Gilman Trail when I lived out here, and this is such an amazing place. It's great to be back. And as Emily said, I'm going to talk about my work at the intersection of machine learning and computational neuroscience. But before diving into that, I just want to frame the type of work that I do in the broader context of computer science and engineering, which is this rich interconnected web of different sub-areas. And I really started out in the upper left, thinking about architecture, operating systems, and as I transitioned to grad school, moved to machine learning. And one of the things that I found really exciting about machine learning is that it really sits at this intersection of so many different areas. I feel like it's such an opportunity for rich interactions across the field. Now, if we dive into machine learning, what do I actually work on in this broad area? Well, I think it kind of breaks down into a few different pieces. So I spend a lot of time thinking about probabilistic modeling for generative processes of complex data sets. And here I'll talk a bit today about the pieces that go into that, thinking about dynamical systems and networks and point processes. I also work a lot on the inference algorithms that go along with these models, thinking about how we actually fit these models at scale to large-scale data sets. And I would say also, one of the interesting things and exciting things in the field of machine learning recently is the advent of new techniques for deep learning, which are really forcing us to rethink, how can we incorporate these new tools into our probabilistic machine learning frameworks? How can we use them to develop more efficient algorithms and more flexible algorithms for inference? Now, the reason that I'm excited about machine learning is because I think it has the potential to really catalyze scientific data analysis. And I think this is true across a whole range of scientific domains: ethology, the study of behavior, healthcare, social science, and text, for example. But really, I'm most motivated by the study of neuroscience. And neuroscience, I think, is a really natural area for us as computer scientists to be thinking about because there's such deep connections between the study of how the brain computes and the study of how we develop computational systems. So, for example, one of the really fundamental questions in the field of neuroscience is to understand how the sensory inputs that we're bombarded with shape the types of computations that are performed in neural circuits, and then how those computations ultimately give rise to the rich repertoire of behavior that we produce, how those behaviors control our motor outputs, how we use them to plan our next actions, and how we learn from past experience. And this moment in time is actually a really exciting time to be focusing on neuroscience because new recording technologies give us the ability to go in and record from the brain at unprecedented scales. So, for example, the advent of brain-wide 3D imaging techniques allows us to go into model organisms and record from hundreds, thousands of neurons, in some cases, all of the neurons in an animal's brain. And on the behavioral side, advances in automated tracking, computer vision systems give us the ability to quantify the behavioral outputs of these neural systems in very precise ways. Increasingly, we're able to do both of these side by side. Record from hundreds or thousands of neurons in awake, freely behaving animals performing complex behavior. And while these techniques allow us to ask and answer new questions in the field of neuroscience, they also pose some very serious computational challenges. The first of these stems from the fact that the data that these systems produce are generated at rates of hundreds of gigabytes an hour. So even just thinking about how we start to get a handle on this massive flow of information is a challenge. The second challenge is that we don't come to this data empty-handed. We come to it with a whole host of prior knowledge, scientific hypotheses, theories of neural computation. But somehow there's a gap between these two. And what's in this gap? Well, I think it's our ability to translate these hypotheses into statistical or computational models that make very precise statements about what variables we observe, what's the data look like, and what variables are latent, what underlies these computations that will tell us something about how these systems work. So the first challenge that I'll talk about today has to do with modeling. How do we build better scientifically informed models for neural data? The second challenge has to do with inference. How do we develop algorithms that allow us to fit these models at scale reliably and efficiently? The talk today will be broken down into 3 pieces. The first 2 pieces will be talking about this modeling challenge and largely putting off the questions of algorithmic work that goes into fitting these models to the third section. So diving right in, what types of scientific theories do we come to the table with? How do we think about neural computation? Well, I think a fair assessment is that many of our hypotheses about how the brain works boil down to statements about types and features of neurons. For example, I'll talk about place fields, which are these amazing cells in the hippocampus that fire when you're in a particular location in your environment. I'll also talk about cell types in the early visual system. So we're all familiar with rods and cones, which are the first layer of cells in our retina. Beyond those cells, we've mapped out the circuit architecture to a number of layers deep into this visual system. And we'll look at different types of cells in the retina and ask how we can explain their firing activity as a result of different types and locations. And there's a whole host of examples of these. I won't go into details on all of them, but things like synfire chains, things like ring attractors that allow us to keep track of which direction we're heading in. All of these boil down to statements about types and features of cells and implicitly hypotheses about the statistical dependencies between the activity of these different cell types. My question is, can we build new tools that will allow us to accelerate the search for this type of structure given access to recordings of the activity of these populations of neurons? Now the challenge, is that what we observe isn't the cell types directly. What these techniques allow us to do is record from the activity of these cells. And neurons, in mammalian organisms at least, will fire these action potentials, these brief spikes of membrane potential, which are what actually convey information from one cell to the next. So we get to stick our microscope into an organism and get a recording out that looks like this. For each neuron, a sequence of times at which that neuron fired action potentials or spikes. What we're interested in are the statistical relationships between these neurons that tell us about the likelihood of one neuron firing given preceding activity of other neurons. And moreover, the different cell types, for example, blue and red here, that may govern the patterns of statistical relationships between cells. How can we build tools that will allow us to reason about these latent cell types and statistical dependencies given the observed activity? Well, I propose that we build a generative model for this type of spiking activity that incorporates these ideas into the generative process that gave rise to the data we collected. And we'll think about this process as consisting of 3 steps. The first way that you would imagine generating population activity for a set of cells is to first, based on your theory of computation, sample different types or features of cells. Then, given those types and features of cells, sample some sort of network that tells us about these statistical dependencies between neurons. And then third, once we have this network, we'll come up with a way to sample spike trains that are consistent with that network architecture. And what this generative picture that I'm showing you here is really saying is that we're making an assumption about the form of a joint distribution that specifies a distribution over spike trains we see, as well as all these latent variables of interest, the underlying network, the underlying cell types. We're making an assumption about how that factorizes into different pieces. And now, what I'm going to do in the next couple slides is break down and make precise what I mean by each step of this generative process. So, how can we think about modeling networks? Well, the starting point is to observe that for any network, we can break it down into 2 pieces. A sparsity pattern, which tells us which neurons have directed connections, statistical directed relationships. And then second, a weight that tells us the strength of that interaction. Now, to make this precise, if we looked at, for example, in this network on the left, the edge from the 4th neuron to the 5th neuron, what we would do is we would look into the adjacency matrix, see that in the 4th row, the 5th column, there's a 1, indicating the presence of a connection. And in the 4th row and the 5th column of the weight matrix, there's a strong positive entry, indicating an excitatory relationship between these two cells. In order to model networks like these, we can appeal to a whole host of existing literature on statistical models for networks.
Sure. Why model the structure and the weight separately? The structure is, just this is the weights, whether the weights are non-zero.
Yeah, absolutely.
Like the form is containing the latter.
Yeah, it's a great question. You could combine these two together and just model the weights as being zero or non-zero. So what we'll see in a couple examples here is that different latent variables could affect the probability of there being a connection separately from the strength of that connection. So we're going to break this down into a spike and slab model, essentially. So now let me give you a few examples of how these latent variables could come into play in determining the probability of different network architectures. So the simplest one, the base case, is to assume that there are no latent variables at play, that every entry in this network is just a coin flip, a Bernoulli random variable. But the story becomes more interesting when we say, suppose that there are different types of neurons, squares and circles here, and that maybe the probability of connection is influenced by the underlying type. Squares are more likely to connect with squares, for example. Alternatively, maybe each neuron has a latent location, a continuous valued position, and that what governs the probability of connection is the distance between neurons in this latent space. We can do this for the adjacency matrix, but we can also do this for the weight matrix in the same way. Weights could be conditionally independent. They could be determined by latent types, squares or circles. They could be determined by distance. Now, as I was saying, there's a whole host of literature on models of this flavor that we can bring to bear in thinking about network architectures underlying neural systems. The question then becomes, if I have a model for networks, how do I connect that to a model for spike trains that I actually observe? And this is where we'll appeal to ideas for multivariate point processes, in particular, something called Hawkes processes. Hawkes processes give us a way to write down distributions over populations of spiking activity. And the way they do so is very intuitive. So suppose that we have a system that looks sort of like this here, 4 neurons connected top to bottom in a chain. The idea behind a Hawkes process is that each neuron has some firing rate that's going to evolve over time. In this case, suppose that there's just a constant background rate of activity on the first neuron. We'll imagine simulating this process forward. Imagine that we get a spike on the first neuron. The definition of a Hawkes process is that every time there's a spike on a neuron, it will influence the downstream rate of other neurons. So, for example, here, it'll add an impulse response to the firing rate of everybody that it's connected to. And as a result of this impulse response, we maybe get more spikes, which then beget more impulse responses again on downstream neurons. And this process will continue in a cascade of activity, giving rise to these correlated spike trains over this population. And we can imagine running this process out in time to generate all sorts of rich patterns of activity in this population based on the underlying network. Now, as I've drawn it here, all the connections are the same weight. What happens when you have different weights of neurons? For example, some stronger than others. Well, as you would expect, the effect of the weight is simply to scale up or down, essentially, the impulse response size, and thereby change the number of spikes that you would get in expectation as a result. That's defining a generative process that goes from latent types and locations of neurons all the way to a distribution over spiking activity. What we want to do is take the spiking activity we observe and go backward, go in the other direction, reason about the underlying network, the underlying latent variables associated with it. And now Bayes' rule tells us exactly how to do that. It says that if you can specify a joint distribution for latent variables and spike trains, then you can use that to reason about the posterior distribution over the latent variables and networks given the particular spike trains you observed. Now, it looks simple enough, but unfortunately, this is actually hiding a fairly hard computation in the denominator of this equation here. And I'm going to talk more about that in the third section. For now, take it my word that we can actually perform these computations and approximate this posterior distribution. What can we do with these types of models? So in the first example, I'll look at these place cells. And here's an example of how these work. So on the top here, I'll show you a movie of a spike train unfolding over time from a population of 45 neurons in the hippocampus. On the bottom, I'll show you the location of a rat as it runs around. And now this is sped up 10 times, so the rat's not really this crazy here. But essentially, what what I want you to note is that there's clearly correlations between the neural activity and the behavior. If after collecting, say, 20 minutes of data like this, we go back and we look and say, where was the rat at the time that the first neuron fired? Well, we would see something like this here. Oops, sorry, wrong way. We'd see something like this. This first neuron fires only when the rat's in the lower left-hand corner of the circular arena. The 14th neuron fires up here, 29th up here, the 39th has sort of a split place field. John?
Are these recordings of like multiple single neurons, or are they just?
No, they're simultaneously recorded with a multi-electrode array.
Holding, multiple neurons in place, as opposed to getting like a population recording in a region, because like, how many individual neurons can we record from simultaneously?
Right. So the recording I showed you is from my collaborators Matt Wilson at MIT. There, they recorded these 45 neurons simultaneously.
So they can hold 45.
Yeah, and this was with a multi-electrode array. Now, new techniques, optical imaging techniques, allow you to get more than that. These are actually relatively old multi-electrode array recordings. Yeah. Good question, though. So this is a remarkable property. This fact that these neurons only fire when the rat's in a particular location, this property is actually, this discovery is what won O'Keefe the Nobel Prize a few years ago. Now, the question is, suppose that we didn't have access to this location information. We just had the spike trains. Could we have inferred on the basis of spike trains alone that they were encoding a two-dimensional location? Well, I'll show you how we can do that with these types of models that blend networks and spike trains. The idea will be that each neuron has a latent variable, a location in two-dimensional space. The model for the adjacency structure, the probability of connection, will be distance dependent. So probability of statistical connections will fall off with distance. And then the only thing we'll assume about the weights is that they're non-negative, so purely excitatory interactions. Now, what I'm showing you here is, first of all, the true place fields on top. And the size of these circles, each neuron corresponds to a circle. The size corresponds to the variance of that true place field. And I've color-coded it just for comparison across these two plots. The bottom is showing you the inferred locations using these network plus Hawkes processes models. And what we see is that, with the exception of perhaps one outlier up here, we do a really good job of reconstructing this two-dimensional field on the basis of these spike trains alone.
Sorry, quick question. But I was looking at the graphs and thinking, actually, they don't seem that similar.
I would say, just look here, right? We have a bunch of blue on top. They cycle over to the greens, to the yellows and reds, to the purples. We can quantify this. I mean, it's much better match than you would get from.
Right.
What's that?
They're loosely similar.
I think they're pretty similar. I mean, we can quantify this by going back and looking at what could you do with something like PCA, for example. And what you'd find is that here you get a much better map. I actually have this in the supplementary slides if you're interested. But you're right. I'm appealing to your qualitative notion of similar here. You're exactly right. One of the points I want to make, though, is that we can do more than just get a point estimate of these place fields. We can also compare posterior distributions and ask about our certainty regarding these latent locations. So here I'll show you this for 4 neurons at a time. These 4 neurons in the top correspond to this distribution over locations for these 4 neurons on the bottom. So again, we get this spatial arrangement preserved. Now, what's also interesting is that neurons that have much larger place fields in practice correspond to much larger uncertainty under the posterior. So we have tools that allow us to go in and interrogate this latent structure on the basis of spike trains and get quantified estimates of our uncertainty in them. Let's look at another example here from the retina. And I'll start by just motivating this with a simple example. So in your retina, you go 3 stages in. Eric here is an expert on this visual system, so he can correct me if I say anything wrong here. But basically, there are dozens of different cell types in the early retina. But 2 prominent classes of neurons are those that are on cells, that when you shine light on them, they're basically laid out in this 2-dimensional plane. When you shine a dot of light on the location of the cell, it'll increase its firing rate. And then there are off cells, where when you shine light on them, they suppress their activity, they turn off. Now, the statistical dependencies between these neurons are going to decay fairly quickly as you go farther and farther apart in space. So we can basically think of each neuron as having a location on this 2-dimensional retinal field and of type on or off in this example. And they'll be wired up like this. What we expect is that the adjacency matrix, if I unravel this system and sort them by off cells and on cells, and then I sort them by their location in this two-dimensional field, we would expect to see sort of a banded structure. It's reflecting the fact that neurons are statistically dependent when they're close by to one another. And then when we look at the weights, we expect to see type-dependent structure. Cells of the same type are positively correlated. Cells of opposite types are negatively correlated. So if we build a model that has this notion of types and locations in this form of dependencies for the adjacency matrix, the weight matrix, can we infer the locations and the types from the spike trains? So indeed we can. Here's an example showing you with real data from a macaque retina, 27 cells, 16 of which are off cells and 11 on cells. That it's a bit harder to see with so few cells, but you get this three-banded structure. And when we look at the inferred locations versus the true locations, which were determined on the basis of the stimuli, again, we find that we are able to reconstruct this two-dimensional map from this neural activity alone. And now here, we can really go in and we can quantify, how accurate are these pairwise distances between cells? Perhaps we can compare this to something like principal components analysis, another way of doing dimensionality reduction on these spike trains. Or, even fancier, compare it to something like a linear dynamical system, which is like PCA but accounting for temporal dependencies. And what we find is that these methods allow us to reconstruct this in a way that wouldn't have been possible with these alternative off-the-shelf dimensionality reduction tools. And now why is that? It's essentially because here, these dependencies on distance only reflect the fact that you have a statistical relationship, not the sign of that relationship. And so this pops out of a model like this, where otherwise would not have. If we go in and we look at the weights, here we see evidence of these latent cell types. We find that there are two clusters of cells. We can separate them exactly. And we can find that cells of the same type have these positive red blocks here. Cells of opposite types have inhibitory relationships, shown in blue. And now finally, you could ask, well, you've kind of cheated. I've cheated a bit in saying that I already know that there are types and locations. I know that they influence the adjacency matrix, the weight matrix. How could we have discovered this a priori? Well, a simple thing to do is just enumerate all classes of network models, all types of latent variables and how they could depend on how the adjacency and weight matrices could depend on them. And what we see is that here, this particular formulation pops out as the best model for this data, giving us a way to explore a whole class of hypotheses about latent structure given spike trains alone. So just to summarize this section, what are the contributions that we've achieved here? We've developed a framework for translating hypotheses about types and locations and features of neurons into models for spike trains. We can use these models and the parameters inherent in them to quantify differences between different populations of cells or between the same populations of cells over time and throughout learning. In these examples, I've held out the sensory stimulus, the light that's shown on the retina. I've held out the location of the rat. But it would be a trivial extension to incorporate that into the models. Here I just held them out for validation purposes. And more generally, these types of models, while inspired by neuroscientific applications, have much broader applicability. So for example, some of the earliest work in Hawk's processes were on studying earthquakes, seismic activity, and thinking about how earthquakes at one location are likely to excite future activity nearby. We've also looked at these as models for crime, for gang-related activity in Chicago, thinking that gang-related activity in one location is likely to beget future activity in the similar locations. And finally, these are very popular models in finance, where people are trying to model the arrivals of different orders for different stocks. But perhaps the most striking application of these models is in social network data. So here, I'll use a toy example from the popular HBO show Game of Thrones. And so if you're not familiar with this show, I'm not going to give anything away here, don't worry. Essentially, this is a really gross simplification, but essentially, this show is consisting of a whole cast of characters that have different allegiances to different families. They're always switching sides. And you could imagine that, suppose you wanted to infer that type of clan structure from some activity. For example, from exchanges on a social network that's linking these characters together. And we can't forget Hodor. So what does this actually look like? Essentially, what we're talking about is a social network where we have a point process unfolding over time for each of these characters, events in time with corresponding metadata, some text that's being sent between them. From this data, we would like to infer some latent network structure, perhaps some latent types associated with these different characters. So let's switch gears now, and let's talk about a second example, here in the context of modeling neural activity in one of the canonical model organisms for neuroscience, C. elegans. And so this is C. elegans here. It's a small worm, and this is a video of it crawling around from my collaborator, Andre Brown. Essentially, it's crawling around, and we have it in a dish that we can track it and keep it under the microscope. And what you see is that every so often, it'll turn directions, and sometimes it'll execute these elaborate turns, just like this one here, where it stops and goes in a totally different direction. The question is, this worm only has 302 neurons. How do those 302 neurons give rise to this behavior? Now, to really study this, what I'd like to do is record from all the neurons in this brain as it freely crawls around. We're starting to get the abilities to do almost that. But for right now, I'm going to instead study neural activity in an immobilized worm. Roughly 100 neurons in the head ganglia of this C. elegans. And here this is with a different collaborator, Manuel Zimmer. So each white dot in this movie is a neuron. This is expressing a nuclear localized calcium indicator, which reflects its underlying activity. And as this movie plays, you can see by eye again all sorts of correlations between neurons in the center here firing together. Neurons up top that seem to flicker on and off in anti-correlated ways. How can we relate the dynamics of this activity of roughly 100 neurons to the behavior that I just showed you? Well, the first step will be to look up in our textbook. Since this is such a well-studied model organism, we have a host of knowledge about this system. We know the identities of each of those 302 neurons. They all have a name. They're all known where they're likely to be found in the worm. So we'll line up that movie with a map like this and try to figure out which neuron's which. And this process will be imperfect. So we'll only get a subset of these neurons accurately. Some neurons, for example, ASKL and ASKR, we couldn't identify them in this first worm. But the hope is that since we can do this across a population of worms, all genetically identical, that in some worms we'll get different subsets of neurons. And from this collection of partially observed neural activity, reconstruct some sort of model for the C. elegans brain as a whole. What types of hypotheses can we use to inform this model? Well, I'll enumerate a few here. The first is that the behavior that this population ultimately gives rise to is basically consistent of a handful of different actions, things like forward crawling, reversals, ventral turns, dorsal turns, etc. Moreover, when we look at the neural activity, we see all sorts of correlation structure here. And what this implies is that this brain state, even though we've recorded 100 neurons, we should be able to capture much of the variance in this signal with many fewer latent dimensions. So for example, here is the observed neural activity. Perhaps we can approximate that as a function of, for example, a two-dimensional brain state unfolding over time. If we were to plot this two-dimensional brain state, not as a function of time, but against one another, this would trace out a trajectory in this two-dimensional space. And the critical hypothesis is that different behaviors, forward crawling, reversals, etc., correspond to different patterns of dynamics in this brain space, this latent space of brain activity. Now, the piece that will allow us to tie together across worms is the hypothesis that since these worms are genetically stereotyped, the dynamics that we find in one worm should be very similar to those that we find in other worms. Let's take these hypotheses and build a probabilistic model for neural activity out of them. We'll do so.
Question, could you say something about how the worm was immobilized and why you think it's trying to move?
Yeah, absolutely. So the worm's actually fixed in this little microfluidic device. It crawls into this space, and then they put a local anesthetic on the muscles in the tail. And so we can't say for sure that the neural activity is trying to create movement, but we can look at which neurons are activated, and I'll show this in a second. Some of these neurons, when they're active, they will deterministically activate certain muscles. And so on that basis, using, again, appealing to prior knowledge about this system, suggests that these are executing behaviors. So again, the starting point is that neural activity is reflecting a sequence of motor commands. These will be discrete latent states in our model, things we don't directly observe. Here, think of them as a sequence of blue, reds, and yellows. The second hypothesis is that these discrete states give rise to different patterns of dynamics in this low-dimensional state space. Here, blue has fast oscillatory interactions, red, slower dynamics, etc. And then finally, what we actually observe is a noisy projection of this low-dimensional brain state. And since we'll be working in discrete time, what we actually will be modeling are sequences of vectors. So here, y will be the neural activity observed at time t, x will be the continuous latent state, z the discrete latent state. And when thinking about the probabilistic model that we're prescribing, we can think about it in terms of a graph that has nodes corresponding to all the different variables here. And the ones that are filled in, these are the variables we actually observe, the neural activity unfolding over time. What we seek to learn about are these continuous and discrete brain states that correspond to different patterns of neural activity. The arrows tell us about the assumptions about different dependencies that relate these variables to one another. So the arrows from the continuous states to the neural activity reflect the hypothesis that neural activity is a function of this low-dimensional latent brain state. This set of arrows here tells us about our hypotheses about the dynamics in this low-dimensional state space. To get the continuous state at time t, we have to know where we were at time t minus 1, but we also have to know the discrete state at time t, since that tells us which dynamics to apply in order to update our continuous latent state. And then finally, we have to have some hypothesis about how we transition from one discrete state to another, when to go from blue to blue, blue to red, etc. Associated with each of these dependencies will be a set of parameters that will sit up top here in this graphical model that we'll also have to infer. So not just the latent states, but also the parameters associated with them. And while that graphical model is making assumptions about what variables are related to what other variables, it doesn't make precise statements about how those dependencies are structured. And so here, we'll start with the simplest forms wherever possible. We'll start by assuming a linear mapping from continuous latent state space up into the space of neural activity. And moreover, we'll assume that for each discrete state, we have a set of linear dynamics. So essentially, we're saying brain activity is generated by switching between different dynamical regimes, each with simple linear pieces. And then finally, we'll assume that the transition structure from one discrete state to the next is parameterized by a simple transition matrix like shown here. It tells us for every entry, which state is how probable is it to transition from one state to the next. Under this parameter, sorry.
You don't get spikes, right? You just get intensity measurements?
Yeah, so, and I didn't say this, I should have said this explicitly. So C. elegans is interesting in that it doesn't spike. So in invertebrates, this is actually relatively common. What you get are these essentially changing voltages, but not spiking activity.
So this is switching common filter?
Yeah, that's another name for a switching LDS. We're going to start with this as our base case. And now we're going to say, well, where does this fall short? So one of the shortcomings of the standard switching linear dynamical system is that it doesn't have any knowledge about how the transitions between one discrete state and the next depend on your location in this continuous state space. So for example, we might assume that different states are active or used in different regions of this continuous latent space. And this motivates a class of models that I've been studying recently, which I like to call recurrent switching linear dynamical systems. And the change is relatively innocuous. It's to add these red arrows that allow the discrete states to depend not only on the previous discrete state, but also on your location in continuous latent state space. And intuitively, what this allows us to do is to carve up this continuous latent space into different regions with different linear dynamics. So for example, the blue dynamics may be used in the upper left, red in the upper right, and so on. And I should say that this is a class of models that's related to ideas like piecewise affine models and hybrid systems in the controls literature. The final piece, though, is to ask how can we connect these across worms? And so this is where a hierarchical modeling assumption will come in. The idea is that each worm will be given its own switching linear dynamical system, its own set of continuous latent states, its own discrete states and parameters. But in order to tie them together, we'll make an assumption that the parameters in one worm are similar to those of the parameters in another worm. And this will be implemented by a shared set of parameters that sit up top. The other key piece to note here is that the effect of having this hierarchical model will be that we'll be allowed to fill in the missing data that we don't directly observe in one worm by looking at the correlations and the parameters that we learn in other worms. Now, again, delaying the actual algorithmic details of how we fit these models until later, let's just look and see what can we do with these models. What types of latent states and parameters do we actually infer? And so the first thing to look at is how it segments neural activity. So here I'm showing you a section of neural activity, 1 minute's worth of activity on the left for one worm. And we see that it's broken down this high-dimensional time series into individual pieces. When we look at the continuous representation that it's learned, here looking at the top 3 dimensions of this continuous latent space, we see that what it's done is it's found a low-dimensional representation that approximates this activity as a trajectory through latent space, and it breaks down this trajectory into different sections, each with simple linear dynamics. But is this a good segmentation? Well, one way to answer that question is by comparing it to what an expert would label on the basis, going back to your question, of the activity of individual neurons with known behavioral correlates. So this is an expert segmentation. This is our inferred segmentation. And when we look at the overlap between these, it's not perfect, but we see a strong overlap between manually labeled states and inferred states. And moreover, these manually labeled states, as I was saying, have behavioral significance. So some of these states correspond to reversals, to slow forward crawling, to ventral turns and dorsal turns, etc. Essentially, we've built a tool to automatically segment neural activity into behaviorally meaningful states on the basis of simple assumptions about the dynamics of brain activity in these different discrete states. Emily?
Does it make sense to assume that all of the neurons are changing the dynamics simultaneously?
That's a great question. The question is, this model implicitly assumes that all the neurons change state at the same time. So could you have adopted a different approach? And I think I've talked to Nick Fody about some of his work on similar questions. You could imagine that there are sort of subpopulations of the brain that are changing state separately from one another. And I haven't explored that in this class of models, but I think it's a reasonable assumption as well, a reasonable hypothesis to test. Now, a key piece of this is that we can apply it not just to one worm at a time, but we can apply it to the whole population and pool information across the population to learn shared states and similar patterns of dynamics. So here, what I'm showing you is how it segments activity from 5 different worms. And what's interesting is that the hierarchical model allows for enough slack so that one worm can be slightly different from another. We can capture little idiosyncrasies between worms while still getting the same qualitative breakdown of states into different discrete and continuous states. Practically, tools like this allow us to fill in missing data. So we can make predictions about the activity we don't see. And intuitively, the reason is that we learn correlations between neurons in one worm. Those immediately apply to other worms as well. But a key question is, did we need such an elaborate model? Did we need all these different pieces? To answer this, suppose that we looked at a model that didn't have recurrent dependencies. It was still hierarchical, but no recurrent dependencies. When we fit such a model to neural data, we see that it again can segment reasonably well. That is, you don't need the recurrent assumption in order to build a good segmentation tool for neural activity. But if you want to use this tool to generate and simulate neural activity in the worm brain, that these models will fall quite short, as you'll see here. So here I'll simulate forward in time from this hierarchical but not recurrent model. And what you see is it doesn't take long before the simulation goes completely haywire. And if I simulate 18 minutes worth of data, you get something that looks like this. So perhaps a good computer art tool, but not a particularly good brain simulator. If we instead simulate from the recurrent model, since it knows not only the transitions from one discrete state to the next, but how they depend on where you are in this continuous space, we'll be able to simulate much more realistic data. Now, there's still room for improvement, but I think that this is a great step toward this goal of building simulators for neural activity. And the true test will be, not only can we simulate this in continuous space, but when we project this back up into the space of neural activity, what we see is that we get very accurate recreations of neural activity for the whole population of C. elegans neurons. So wrapping up, what are the practical consequences of this work? So for one, having a generative model allows us to perform what are currently very tedious and laborious manual tasks. So for example, if we can simulate neural activity, then we can use that as a tool to help us aid in this very first step, which is identifying which white dots are which neurons. So we can make predictions about what neurons should be doing, and on that basis, we can help to automate this first step of the process. The second piece is just as before. Having models like this gives us interpretable parameters that we can go in and we can ask, how do these parameters change between, for example, different groups of worms? Worms of different genetic strains, worms at different developmental stages of life. We've started to do this in collaboration with Manuel Zimmer. Finally, while the worms were immobilized in this case, having a model for this brain activity suggests hypotheses for the dynamics of neural activity in freely crawling worms. And I'm part of a collaboration now with Andy Lafer at Princeton, where we're using his new techniques for recording neural activity in freely crawling worms to start to get at this question. Finally, and this is more of a theoretical question, the models that I've developed so far are based on these ideas of latent states of neural activity. What we'd really like to do is tie these ideas of states back down to some underlying biological mechanism, really understand the basis of neural computation in C. elegans. And what I haven't told you is that not only do we know the names of these neurons, we also know how they're all connected to one another. We know the connectome, so to speak. So a question is, if we know the adjacency matrix, the connectome essentially, can we find weight matrices that are consistent with that connectome that would generate switching activity like what I've shown here? And this would really help us to tie this long-standing goal of neuroscience from mechanism up to observed neural activity. And finally, just as before, these types of tools aren't just useful for neuroscience. Essentially, these are tools for modeling nonlinear dynamics and breaking them down into simple pieces, simple linear regimes used in different sections of this continuous latent space. And this has applications across, I think, computer science as well. So think about power systems, for example. These are complex systems, and while they're white box, we can look into them. The phenomena, the patterns that they generate are extraordinarily complex. Having models like these could help us break down and think about these complex systems in terms of simpler bits. Yeah?
So, but why not just use the known structure of the network and just train the weights? How would that do compared to your model?
Yeah. So the question is, why not just train a network model if that's what you're ultimately after? So one reason is that there's many more weights in this network. There's roughly 7,000 synapses. So there's a lot of parameters that you would have to tie down. A second is that...
I would call 7,000 a lot.
Well, remember that we're only dealing with roughly 3,000 time bins here for each neuron, for each worm. So we are in a fairly limited data regime. Now, your point's well taken. And I think that that's actually a next step, is we want models that ultimately get us toward this question of the weights of interacting neurons. But I think we also want descriptions that exist at different levels of abstraction. So not just the weights, but also this more intuitive description in terms of different brain states that correspond to different behaviors. So the last section, and the shortest section, will be on the algorithms that we actually have developed in order to fit models like this at scale. And in order to reason about the challenge of Bayesian inference, we don't need to think about the big graphical model that I showed you before. We can talk about it in terms of a simpler model, one that just has observed data y, some latent variable z, and some parameters theta. The goal is to approximate the posterior distribution, which, as before, we said is given by Bayes' rule. And as I alluded to, the challenge is that the denominator in Bayes' rule is hiding this nasty integral, an integral over the space of parameters and latent variables. So in order to approximate this, which we'll have to do for any non-trivial model, there's sort of two meta-algorithms that we can appeal to. There are Markov chain Monte Carlo algorithms, which will try to design stochastic processes to sample from the posterior distribution, random walks over the space of theta and z. And there are variational inference algorithms that will try to approximate the posterior with the best in a smaller and more tractable class of distributions. For example, the class of multivariate Gaussians. Now, I'm not going to go into all the mechanics of how these work, but in both cases, we can appeal to a similar form. The idea will be to exploit the structure of this graphical model to make updates of this posterior distribution more efficient. So these will unfold in a sequence. The first step will be to update big chunks of variables at a time. Update, for example, all the continuous states across each worm, holding fixed the data below and some initial values of these discrete states. And then, conditioned on these particular continuous latent states, proceed upward and estimate the discrete states given them, and then percolate up all the way to the top until we get to these parameters that are shared by all worms. And at this point, our estimate of these parameters will be informed by all the data in the entire data set. So when we proceed back down, updating these variables in reverse order, now these continuous states will be informed not just by the data below, but also by our parameter estimates that were informed across the entire population. The challenge is that, unlike in the standard SLDS, which doesn't have any of these recurrent dependencies, in this case, the model was nice and easy in the sense that the conditional distributions over these continuous states were all nice and Gaussian. When we go to the recurrent case, what we find is that it's no longer Gaussian, it's actually tilted or modulated by all of these discrete dependencies that are hanging off of them. And what this means is that we can no longer apply our nice efficient tools for sampling from linear Gaussian models. So how can we overcome this? Well, one class of approaches that I've been working on are augmentation schemes. And the picture is sort of like this. Here's the one-dimensional reduction. In our true model, we have something that's like this blue curve, not quite Gaussian. The red is the best Gaussian approximation to it. The insight, or the idea, is to do something that's seemingly counterintuitive, to take this one-dimensional problem and turn it into a two-dimensional problem. Now, while this seems like it's only making our integration problem harder, it has the nice property that if we marginalize, we recover, that is, integrate over this y-axis, we recover the true variable. And if we take slices, horizontal slices through this, we get exact Gaussian conditionals. And so what that means is that we can get back this nice Gaussian structure, and we can use all of our nice efficient inference algorithms for linear Gaussian models. Now, these augmentation schemes apply in only a limited subset of all possible recurrent dependencies. What if we want to incorporate things like more complicated nonlinear recurrent dependencies that would carve up space in arbitrary ways? For example, by incorporating neural networks into these graphical models. So to give you a sense of the types of work that I've been doing very recently and that I think will extend into the near future, what we're trying to do are build up better inference algorithms for these class of models that blend probabilistic graphical models with neural networks. And the idea is essentially the same, that the problem hasn't changed. We no longer have Gaussian structure. What we'll do is try to approximate these non-Gaussian pieces with nice Gaussian dependencies. Another line of work that I've been exploring, and that I won't go into much detail on, is to ask, suppose that we wanted to do away with this switching linear thing altogether. Maybe go to our network model like you described. This model would have complex nonlinear dynamics associated with it. Could we still develop variational inference algorithms to target the posterior of these nonlinear dynamical systems models? So one class of work I've been doing is to take ideas from variational inference and blend them with ideas from Markov chain Monte Carlo in order to develop more flexible variational inference algorithms. And I'd be happy to talk about this more offline if anyone's interested.
Question.
Yeah.
Why not just use an AP inference, which would do away with this intractable integral and all the approximations that you have?
Yeah, so we've talked about this a little bit. The question is, why not just do away with the whole Bayesian thing and find perhaps just a point estimate, maybe the most likely assignment under the posterior. So, one challenge with this is that the likelihood of this, the marginal likelihood, is this very non-convex objective function. Now, certainly, the experts in deep learning have come up with better ways to, well, at least empirically, better ways to try to tackle these non-convex problems. It's something we're actively exploring. Now, I think there is still some value to trying to approximate the posterior, but I'm also willing to concede that in many cases, we're kind of after sort of a point estimate, a breakdown of states, and less interested in that variability. So it's a fair criticism. In the last section here, just to wrap up, I want to talk about, where does this go from here? What's next on the horizon? And over the short term, I think it's fair to say that neuroscience, if it's not there already, it's very clearly headed toward a domain where high-dimensional data from multiple modalities at once is becoming the norm. And so to give you an example of this from collaborators at Harvard Med School, this is a data set that we're working on where we have a Kinect video of a mouse running around in an open field, and then this cable that's protruding out of its head is collecting a video of neural activity in the striatum, which is a region of brain that's involved in action selection. And what's clear already is that if we're going to get some insight into how neural activity gives rise to behavior, we're going to have to break down these two very complicated different domains in order to come up with relationships between them. And I think that this type of data is by no means constrained to this one example. That there are all sorts of examples of labs here at the University of Washington that are collecting exactly this type of data and facing exactly these types of problems. So for example, you may have heard of the new Center for Computational Neuroscience, which is a new initiative here that is really, that Eric Shea Brown, I think, is very critically involved in. That this is trying to bring together people to think about these types of challenges. And the first line of their mission statement is essentially the type of work that I aim to do. Develop the computational and theoretical tools to help interpret this extraordinarily complex data, not only generated here at the University of Washington, but more broadly in Seattle. So just down the street, the Allen Brain Institute is leading the world in generating large-scale neural data, not just neural recordings, but also behavioral data, underlying structure like the connectomics, the connections between neurons, the genetic expression data, the morphology of cells, all of this data coming together to give us these huge data sets spanning many different domains. How can we build tools to bring these all together? The challenges that these types of data pose, I've talked about two of them, modeling challenges, inference challenges, but there are all sorts of places where computer science can make serious contributions toward this goal. There are challenges related to how do we even build scientific tools that allow us to criticize different classes of models and search for the best models for particular data sets. There are engineering challenges. How do we process these massive streams of data? Experimental challenges. How do we design stimuli to probe these systems in the most informative way? And more on the theoretical side, how can we use what we know about developing computational systems to inform our hypotheses about how the brain works? By way of conclusion, I want to end with what I think is a nice unifying view for how I've been trying to approach these problems and how my work is trying to address them going forward. And I think a fair assessment is that as we're trying to tackle scientific data analysis challenges, we have a host of tools at our disposal. And these tools can roughly be broken down across an axis of two competing features. One, their ability to give us interpretable descriptions of complex systems, and two, their ability to flexibly capture the complexity of these systems. So to give you some examples, high on the interpretability axis, perhaps low on the flexibility axis, off-the-shelf tools like clustering, linear dimensionality reduction. And certainly, the advances in the field of deep learning are pushing out on this flexibility axis, but often at the expense of giving us interpretable descriptions. And I know clearly around here, there's a lot of work at trying to make these models more interpretable. And certainly at all these points in between, there are models that trade these off in different ways. But in some sense, there's a frontier that holds us back. And what is the source of this frontier? Well, first, it's our ability to articulate models that are both flexible and interpretable. And second, it's our ability to then go and fit these models to large-scale data sets. And my work has been about pushing this frontier up and to the right. I talked about Hawk's processes and networks, talked about recurrent state space models, I talked about new inference algorithms. These are the types of work that I'm going to pursue going forward to address the scientific challenges that are present in neuroscience today. So with that, I'd like to thank all of my experimental and scientific collaborators and to thank you for all your attention.
questions? Yeah.
Pedro.
So clearly there's a lot of use that, computational science can make use of models for machine learning, but there's also a lot of benefits that we potentially can gain machine learning by learning from neuroscience. So my question is, what have you learned from all this, from the neuroscience that you've been doing, that could be useful in machine learning? For example, as a source of new model, or as a source of new understanding of behavior, etc.
Absolutely. Yeah, I think I really focused on one direction of a two-way street. I focused on how we can develop new machine learning methods to analyze neural data. But I think that, as you were getting at, there's a lot that we can learn going the other direction. So there's sort of two aspects to this in my mind. One of the aspects is that the problems that neuroscience pose suggest new classes of models. And I showed you some examples of that today. Models that bring together networks and point processes, models that force us to push the boundaries of state space modeling in new creative ways. And these have very broad impact. I talked about examples in social networks, nonlinear dynamical systems. I think there's a whole host of ways in which that could drive machine learning forward. And I think part of your question was also, though, the brain is ultimately a learning device. How can we learn about the algorithms that it's using and use those to inform better models in computer science and machine learning? I think we have fewer examples of that really translating, but that's certainly the promise that we're trying to realize. So, reinforcement learning is perhaps one of the best examples of where we thought about these problems from the field of psychology and then informed new machine learning algorithms. But there's a lot more to be done there. Yeah.
It seemed like a lot of the examples you gave were cases of the most dense understanding and data collection that we have in neuroscience, and then you're able to show and validate your methods, which is a great and important step. But you would think if you take a neural pixel probe today and you stick it in the brain, although it has, 1,000 or whatever pound is, they're spread out in a line. The chances that those neurons in that line are all the ones together doing the same thing, like they're selected together place cells or the selected population, is very unlikely. So do your technique scale to cases where you really have a sparser sampling, which is really what we still have in most really unknown systems, and how has it taught us things in those systems, or is it still too fragile that you haven't taken it out and said, okay, let me get people's sparser recordings from higher cortex, where we don't know that there's on and off cells. So where are you with respect to really deploying this into the true unknown and not staying back where you know?
Yeah, I mean, that's a really great question, and it's getting at, I think, some of the really fundamental questions in neuroscience related to what can we learn about neural computation when we observe such a sparse sample of all neurons. That was part of the question. The other part of the question was, what have we learned from applying these methods to neural systems? Now, the first section of the talk was on using this combination of networks and Hawk's processes to study systems. And as you were saying, certainly our first goal was to validate that we can indeed recover the correct answer in certain cases. And we've been applying that to more complex systems, and that's still work that's under development. The second part of the talk, however, was about developing new tools for studying computation in C. elegans. And there we really have, I didn't have time to go into it all today, but we really have started to come up with new discoveries about the dynamics of brain activity in these systems and how they may relate to behavior. So in particular, we've been able to go in and quantify the behavioral dynamics and use these to really ask questions about, is our manual segmentation of behavior correct? Or based on the way that these inferred segmentations have come out, are there nuances or differences in these dynamics that aren't apparent by eye? So yeah, I think obviously this is an ongoing process of developing new tools to solve neuroscientific problems, and then the longer term process of actually applying those and coming to new scientific insights. I mean, these are all things that there's a whole pipeline, and these two are influencing and reinforcing one another. Now, to come back to your question about, is this whole exercise in studying neural activity pointless because we can only see such a tiny fraction? I think, the field of neuroscience is really trying to come up with new ways it's trying to think hard about that problem. And so there's some nice theoretical work, for example, from people like Surya Ganguly, who are saying, what can you learn about the dynamics of a system from a partial observation? And here we have some theories that allow us to really say that, yes, we're not just, modeling essentially noise, that we can actually extrapolate and expand from the limited views that we do have. So yeah, there's answers at a variety of levels there. Thank you.
More question, Sean?
This is more a question about your perspective. Related to the last question, but suppose it's not so much an issue of the sparsity of the sampling. It's that, like in this hippocampus study, I mean, it probably will work there, but suppose you didn't get recordings of where the rat was wandering around, and you just took some multi-electrode recordings, and the complexity of your latent variable model might have to be very, very high because we don't know what's happening in the world, but if I knew this thing was searching for food, then all of a sudden everything would look much simpler. And so once you get to higher level functions, somehow unless we get the data and kind of know how to get these outside world information, how do you get at, are we just building these very, very complicated models due to the fact that we just aren't recording the right exogenous variables?
Yeah, that somehow the story would become so much simpler if we just conditioned on the right thing, right? Yeah, I think this is really what's motivating the shift in neuroscience away from really highly constrained simple stimuli, simple behavioral tasks, toward actually asking an animal to perform more naturalistic behaviors in the face of more naturalistic stimuli. And the hope here is that in this setting, we can record behavior in a very generic way. If we can put a Microsoft Kinect camera overhead, we don't have to throw away all this subtle information about pose and just model the mouse as a point mass. We can take essentially the highest resolution measurements that we can of this behavioral output. And the hope, as you're getting at, is that somehow in that process we'll keep as much information as possible about the types of outputs that these systems are producing. Now, you're right that we're never going to know for certain that we haven't thrown away some key covariate. But really, this is, I think, what neuroscientists are aiming to do is collect as much information as they can and use that to build and constrain better models for these systems. So, yeah, we're working on it, but it's absolutely a hard problem.
Okay, well, let's thank Scott and take more questions offline.